Yes, sir, your screen is visible.
Okay, thank you for confirmation. So, hi, good evening, everyone. So, today's topic is like signal detection and analysis, or you can say basics of the signals. So, any idea what is signal? Like, why we are called as signal and what is signal? Any idea? So, when we are saying signal, so signal is something like when you are going through the road, then you get so many signals, that green signal, red signal. So same way, and every signal have their own definition. That red signal means you have to stop. Green signal means you can go. Yellow signal means you have to just have to wait. Okay, or you are about to wait because that yellow became red after some time. So same way, we have some signals in PV as well. So what is that actually? So signals are something which was previously unknown or incompletely new. Okay, if it is unknown, like it is something new, PV industry, then it will be a signal. But something which is undefined, or you can say it was incompletely known previously, now we come to know some new aspect of the previously existing criteria, then it would be a signal again. So check now what is what is a signal? Information that is arises from one or multiple source which suggest a new potential causal relationship or a new aspect of known association between an intervention or an event or set of related events, either adverse or beneficial. Okay, so this is actually the various definition of signals based on the various, you can say various regulatory authorities or various those the global leaders, like some definition is from ICH, some definition is by WHO, some definition is by UMC. So these are like definitions which are given by the various institutes. Okay, Pisha, can you mute yourself, please? Pisha Taylor. Okay, so first is like what it's saying, information that arises from one or multiple sources. So whether the information which is from received from one source or from multiple source, which suggests a new causal relationship. Like previously, it was not known that the paracetamol is useful or is causing something this. Okay, or maybe they have the causal relationship between XYZ event. But now it is proven that paracetamol is have a causal relationship. Whenever we are taking paracetamol, it is causing us an XYZ event. So, and whenever we are not taking paracetamol, we are not, getting experience with the XYZ adverse events. So this would be a new kind of aspect or association. So this might be a signal as per Sion's law. So what is other like David's textbook of ADR is saying regarding signal? So new hazards or known hazards that is quantitatively or qualitatively different from the existing expectation. So yes, like what was previously known, and it was the previous expectation that is completely changed with the same event or with the same hazard. Okay, or it may be new hazard or it was previously known hazards, but the quality and quantity both is different from the existing expectation. Then it is also a signal. As per WHO, reported information on a possible causal relationship between an adverse event and a drug, the relationship being known or incompletely documented previously. So that is the standard definition which we follow in PV field actually. That is like reported information on possible causal relationship between the adverse drug and adverse event. With you can say the causal relationship being unknown previously, or it was incompletely documented previously. And now we are exploring the new era, or you can say new chapters of those known causal relationships. Or what about UMC? What UMC is defining? So UMC is saying a signal is a hypothesis together with the data and argument, but is not only the uncertain, but also primarily in nature. So signal cannot be, you cannot conclude something very certain with the signal. Signal is something very primarily, examination or evaluation of condition. Might be it is an invalid signal. When you go in depth, you evaluate more, and you are drilled down and narrow down the filter, or you narrow down the criteria, then you come to know that no, it is not qualifying for the valid signal. So it is an invalid signal. So it might be possible, but the based on the primary evaluation or the primary examination, it is known as a signal. Okay. So these are various in definition of signal based on the various, you can say, the institutes, the global institutes like UMC is there, WHO is there, this textbook, David's textbook, or from Sions, which have defined their own way of the signal. Okay. So what is more about signal? A signal in pharmacovigilance is an statistical association. Okay. It is not just a signal, but it is something statistical association. It gives you an stats like from paracetamol. If you are saying nephrotoxicity was was unknown completely. Now we come to know that there is an effect of paracetamol that it causing nephrotoxicity. Okay, or it is damaging your liver or kidney. Then how many reports are there? We have complete stats for that. So we can, in PV, as you know, we are all here using databases. So you can search paracetamol and nephrotoxicity. And even also you can have an idea of age group, like along with genders. Like if you are getting 10 report, out of that 10 reports, all reports belongs to the male patient. So it means female patients are still excluded from this signal. Okay. Once we come to know that this nephrotoxicity is only on male patients, then it will be new aspect. Okay. Or once after 5 years or 10 years down the line, it is like reported by female patients also, then it was again something which was previously not known, because previously it was defined that PCM is causing nephrotoxicity to male patients only. In previous signal, it was defined that PCM is causing nephrotoxicity on male patients only. Female was untouched for or not defined anything regarding female patient during this signal evaluation. But after 5 year, 10 year down the line, if we come to know, no, female patients are also getting nephrotoxicity because of PCM, then it will be new aspect of the signal. Okay. So it is kind of statistical data. It will giving that from initial only evaluation, you can come to know that it is causing to the male, and that male is whether in adult group or whatever they are from the geriatric patients, or you can say the adult or the adolescents people are facing this kind of events. So that is actually a statistical association, because you get full stats. It consists of a hypothesis together with data and arguments in favor and against to the hypothesis. So yes, what it means that it is an hypothesis, means it is something, you can say, examination. Hypothesis means examination or hypothesis means your own what you say, which you have explored. No one else have explored it. Okay, so it is your own hypothesis. Okay, so it may be like it is a hypothesis along with data because you have data. Like as in the previous example, I have told you that PCM is causing nephrotoxicity. If you can come to know that PCM is causing nephrotoxicity and to 10 male patient, you can argue in favor of that if you got those data. But if you do not got any case from the PCM and nephrotoxicity, then you can also comment to against to that hypothesis as well. Okay, someone is published that PCM is causing nephrotoxicity. But in case if you are not getting any data or you are getting so many data, then it is something against. You can say, no, it is not known. It is not unknown. It is already known, and I have the bunch of data in which it is mentioned that like if you have hundreds of report in your database, in your repository, then you cannot say it is a signal. Na? Guys, getting or not? Okay. So like what is next? The discovery of the drug-induced disorder from the earliest suspensions via a credible signal to a fully explained and understood phenomena is a lengthy process. So yes, what it is saying that if there was an early suspension of a drug or because of any like there was a drug that is called pioglitazone. Okay. In 2015, it was given to the for the type 1 diabetes to the patients, but there was a complaint that it is a very carcinogenic. So several batches of pioglitazole, which is already present in the market, they are getting, pulled back from the market because of a complaint that it is a have carcinogenic effect. And there was a one patient who got died in the Medanta Multicity Hospital in 2015 itself. So at that time, it was evaluated at signal. Okay. But later, it was concluded no, it was not a signal because pioglitazole don't have any carcinogenic effect, although that patient itself have certain comorbidities. Because of that, patient faced the carcinogenic effect or became carcinogenic, and it was not the effect of that pioglitazole drug. That is basically for diabetes. It is not causing any cancer. Okay, so that was clarified by the MAH, the manufacturers, and after clarification, there was several tests gone through those. Okay, and when they passed those tests, like those certain trials were conducted on the healthy volunteer, on the diabetic patients, and once that the regulatory bodies got full data that it is a safer and what we have uh, revoked previously, sorry, what we have, yes, like drugs removed from the market, they have again, pumped those batches of drug into the market along with certain labeled quantity. Okay, that it is contraindicated with the patient which are having the carcinogen. Like, means patient we have which are already suffering from cancer should not take the the pioglitazole drug because it may cause or it may be life threatening for them. Okay, so that's what I was trying to explain, that how it is like give you early suspension of a drug, which was again once the phenomena was understood and explained, it is again because the MH who have uh, manufacturing those drugs, there is a dent in their image. Okay, why it is a dent in their image? Because it was revoked from the market, and certain batches of a drug when revoked from the market is a kind of negative use, and it is giving a dent to your image. Okay, but apart from that, that manufacturer try to go to the depth study, and they have prepared all the data again. Okay, and after once they get all the sufficient data, they have again put those data in front of regulatory authorities and told them whatever we called as signal previously is not a signal, it is something comorbidity of the patient, and it is not signal. At that time, considering the safety of the public, we have you know taken back those batches of the drug, but now we have full proof data that our drug is completely safe for public. Kindly allow us same batches to pull back to the market. So that's how the regulatory agencies, they have taken back their revoke. Okay, they have revoked the ban, and those batches of the drug is again pumped into the market. It what is next line? It may take years until the symptoms, frequency, mechanism, and risk factor of an adverse reactions have been fully recognized, and causal association, causal connections has been definitely established. So same what I have explained already in the example of pioglitazole, which is a real-time example of pioglitazole, okay, which happens in 2015. And based on that, there was a dent in the image of the manufacturer, but still, without caring the dent on their image, they are trying to, get rid of them, and they are trying to prove themselves that what they have manufactured is completely safe. Okay, a signal is more or less stronger specification suspicion. Therefore, the signal need follow regard of both scientific credibility or clinical and regulatory relevance. So yes, what if the manufacturers just like regulatory bodies told that pioglitazole is causing cancer to the diabetes patient who is taking it? What if manufacturer just accepted it and they do not take any follow up and do not, you know, evaluated their drug again? So that drug ban will be a for lifetime. But they have taken certain follow-ups with regulatory authorities that and they have provided the data again and again. Based on that, for based on that, they are able to prove that whatever they have manufactured, it is not. So they have certain regulatory relevance, it have certain clinical relevance, and they have that data of clinical once they have to prove themselves. Okay. So signal management. So when you are saying signal management, so it is a set of activities to be determined whether it is a new risk associated with any of the active substance or medicinal product, or whether known risk have changed based on the examination of individual case safety reports, aggregate data from active surveillance system or studies, literature information, or other data sources. So yes, it is already I have mentioned if what is signal management, how you have to manage the signal. First, you need to know First, you need to know how is it a signal or not. If yes, then what kind of signal, whether it is a new risk or it is a previously existing risk with new aspect. Okay. There are certain databases, or there are certain regulatory authorities who are always being informed for any of the signal to the any of the country. One is the Uppsala Monitoring Centers. It also required whatever the early signals which are detected by any of the country that need to be reported to the WHO database through with the help of SALA monitoring centers. So that's why, because at country level you cannot conclude anything, but at global level you need to conclude well. Might possibly which something which you are facing in India, it is not faced by some other country. So it need to be cross verified with other continents and other countries all over the globe. Okay, it is very important for the verification of signal. Guys, okay. So that's why WHO database contains like more than 2.8 million spontaneous report ADR, which was reported every quarter. This is the data which they are getting every quarter. Means in a year they are getting what they are getting. They are getting more than almost 10 million reports. Okay, and based on that, um, identifying the signal is very tough job actually. Like 10 million reports per year, 2.8 million report every quarter. So there are certain databases which are available. With the help of that, you can run the query and you will get a verified signal. Okay. So the database is scanned for the ADR, that is drug and ADR combination, and filter out using the different criteria like for which country we are looking for which ethnicity, for which age group, for which particular gender we are looking for. If we get that something, then it is not a date, not a signal. And if we do not get anything, then might be it is a signal. Okay. So the management process for a signal. So these are the various steps which are involved in the signal management process. Like first is the signal detection, then validation, then analysis and prioritization, assessment and recommendation for action, if any, and further, if you validated and each and everything is recommended, then yes, that information which you have regarding the signal that need to be exchanged further. Okay. So I will define each and every step in detail as well. So in database. Okay. So signal detection, it is already that if some aspect is incompletely known or it was known, but this is something new regarding that previously known thing. But you, as I told you, you need to verify saying is it correct or not, or it is just an assumption. So you need to check in literature that you need to do literature searches like for your M.Pharm thesis, your B.Pharm thesis, you must done the literature searches. Same way here, we have check for the if there is any published article for what you are looking as a signal. Like for example, same what I quoted 5-metazole is cancerous for the diabetic one type of patient. So we need to check whether this kind of studies are published by any of the author, by any of the trials or not. If studies are there, but no, but nothing was experienced, okay, then again you need to verify. How you will verify? You have to verify by giving those drugs to certain volunteers. Okay, what we are saying that these are certain blinded studies, because sometimes in patient in when patients are admitted into the hospitals, there are so many studies are running inside the hospitals, even which patient is enrolled into the which studies, that patient itself don't know, but those are known by the hospital authorities. So at that time, when they are part of any clinical trial also, this signal validation can be validated. Like, for example, if in hospital there was a patient who is already suffering from pioglitazole, sorry, suffering from uh, that diabetes 1, type 1 diabetes. And based on that study, they can, you know, administer that pioglitazole drug to those patients and check the effect, because it is already in the hospital and everything is getting observed. So based on that also signal can be validated. But it is harsh reality that patient don't know about that it is a part of validation program as well. Because if that is not confidential with the patient, patient will never enroll themselves. Okay. So those are mostly the blinded studies, or more or less those are double blinded studies in which signal validations are taken care. Once you validate that yes, this may be a valid signal, then signal analysis and prioritization. Further analysis can be done that if you willingly in giving some drug, like every criteria is there with you. What I have told you in the PCM example, that PCM is causing nephrotoxicity in only male patient with specific age group, like from 40 to 60 years age male patient are getting nephrotoxicity while taking paracetamol or nemoslide. Okay, like Nemoslide is in USA, Nemoslide is banned for periodic patients, like people who have age group less than 7 years. They do not take nephrotox. They do not take the Nemoslide because of the risk of nephrotoxicity. Okay, why? Why it was known side effect with Nemoslide that it may cause nephrotoxicity with the 7 or less years of the patient. So it is known. So if it is already known, so you can do the reverse analysis, that whether if I am giving that patient having the 9 year age group, whether they have experiencing something or not. Might be they will not get that much threshold, but they will also get impacted. So you can analyze based on that. And based on analysis, like if it is life threatening signal or it is like not life threatening, but it is still take immediate attention. So based on that kind of thing, you can prioritize. Okay. Like if it is life threatening, then we need to prioritize first. If it is not life threatening but may cause health damages, then also it need to be prioritized. But when you are comparing the life threatening and health damage, so in that scenario, the life threatening would be taken care first, then health damages will be come into the picture. Okay. Signal assessment, recommendation of action. So once you come to know that this is the signal assessment, what is your recommendation? Like if pioglitazole is causing, so what regulatory authorities recommended previously? They have taken all the batches from the market, okay, and they have stopped manufacturing the new batches as well, because they want to get assured whether that drug has certain associated risk of uh, cancer or not. If yes, then they will stop marketing, that drug will get completely banned. But if not, or it has some contraindication, so that drug can be marketed along with some certain label warning, some certain alerts. Okay, and once that is like if regulatory authorities are taking certain alerts, so that information need to be exchanges, and how it will be exchanges, it need to be published into the articles, into the newsletters, into the media. Okay, or even into the mm, doctors need to be get educated so that they will also not prescribe those medicines. Okay, because it is already contraindicated and it is already banned. Okay, because if they are still prescribing those drugs, so might be possible some of the sellers have the older batches of those medicine and they can sell it out. Okay, so that's why exchange of information is very important. Next, significance of signal management. So like we have talked about signal so much, but what is the significance of that? No one is saying that really. Why signal is important? Because if it is, it will give you early warning. Okay, any new ADR or new drug. Okay, like as I told you, it is a preliminary examination or preliminary evaluation of a drug. So yes, if we can conclude something that it is a signal, so it is a kind of early warning. Okay, and we can try to minimize it, or if it cannot be minimized, we can stop that thing there itself if it will get detected in their early phase, right? What is public health issue? Importance, important drug like seriously indicated indication, widely used, serious reactions, large number of cases, rapid increase of in reporting, regulatory actions. So all these steps are part when you come to know that it is a signal. Okay. Then you need to know that what is the important, like for example, what happened in the thalidomide trial thing. Okay, people come to know that it is causing child deformity. Okay, it is so, but clinical trial was there, which we have certain evident data that clinical trial was performed for thalidomide and certain other studies were conducted for thalidomide, but still, so once it was happened first time, at that time it was signal, but now it is a known risk. Okay, change in benefit and risk ratio. So yes, like in the thalidomide example itself, previously it was safer for use. Okay, even for pregnant ladies as well. Now we have a contraindication, or we have a alert warning that it should not be taken with a female who have about to conceive or who have their third trimester of pregnancy. So like that completely shift the benefit risk ratio. Previously, that thalidomide was completely safer for the use for each and everyone. Now we are saying that it should not be taken with the um, patient who are in their last trimester or who are about to conceive. So it completely changed the benefit risk ratio. Previously, we have saying that it is completely safe, 100% benefit. Now we are saying it is so much contraindication. This thousands of people are suffering from the media, and so it is so much risk. No? That is the example that change the benefit and risk ratio. Scientific and educational value. As I told you that once it was uh, identified that it is a signal. So for to get validated your signal, you need to have that scientific evaluation. You need to go the literature searches that whether something written regarding that in any of the article or not. Okay. and not if it is not incompletely known, then it has certain educational value because we have to spread the education, we have to spread the awareness for that signals. Okay, strong evidence. So yes, as I told you, what happened in the example of pioglitazole, that previously some patient or doctor is saying that it is completely because of pioglitazole. They have certain strong example. They have even autopsy report in which it is showing that pioglitazole was in the body of in the traces of pioglitazole was found in the body of the patient. But because of that, the drug is banned. But that manufacturer keep evaluating and they have collected the strong evidence. Based on that, that ban get revoked and the batches of the drug is again pumped into the market along with certain labeled wording. Okay, I believe. So sources, what are various sources from where you can get a signal? So these may be all these are the sources, like first the preclinical data, like preclinical data is something which is called as the animal studies. Before doing the clinical trial is basically on the human subject, and preclinical is something which is before the starting of the clinical trial. We have to perform same study into the animal. Those have the same action with that drug. Okay. Now, those are also a source from where we can get signal. Clinical trial phase 1 to phase 4 till phase like whole clinical trial are the good sources of the signal. Spontaneous reporting, that company database, regulatory database, anything which we have seen that whatever the signal which we find, we need to commit those to the WHO as well so that they can have their global databases. Okay. We can also get the signals from the published literature. As I told you that whenever we have to verify any signal, we have to check whether something is already published for those. Do we have any literature article for those those signals which you need to claim as a signal or which you need to checking, verifying or validating comparator data. So yes, comparator data is also a source of. So what is comparator data actually? For every clinical trial, we have certain set of standard data. Okay, so those standard data and there is some comparator data that first we need to check with the comparator, and once comparator gives you the result, then that comparator becomes the standard and your drug, the test drug will be the get tested again with the that comparator. So that comparator data is a kind of a standard data while doing the clinical trial. So we can check signal in comparator data as well. That comparator data, like comparator drug is this one, and it was not giving this effect, but our drug is giving this effect. So the effect may be a signal from our drug because this was not known with the comparator data. Okay. So that's how we can compare. Observation in individual patient and populations. Yes. Case control surveillance. So case control surveillance is nothing like when you are having control group. Control group means that specific study is going on. Like if it is a diabetic study, so that patient will take only diabetic drugs, okay, or they have certain diabetic conditions itself. So those are case control surveillances, and if those are also like those are taking for their drugs for certain specific criteria, like as I told you, diabetes. So still they can have something new with their own drug, whatever they are taking as an anti-diabetic. So follow-up studies is very important to verify the signal whether it is valid or invalid. Okay, there is no minimum number of cases required to define signal. Signal depends upon seriousness of the event and the quality of the information received. So yes, as I told you that four criteria is important as a valid criteria for an ICSR. But might be possible if one criteria is missing and there are certain other information with that drug and we are claiming it as a signal, then it is an invalid signal and because invalid signal because we are claiming something which is already invalid report because that report do not have that four criteria. Okay. And how can we claim such report as a something valid signal? Okay, and signal is something you cannot define it with a number that we receive one report with this, we receive 10 reports with this event, so it is a signal. No, it is a set of information, or you can say statistical data, which I already told in previous slides. So what is this? This is signal generation methodology, the manual method. So there are two type of methods: signal detection, signal generation, manual method, and there is one automated method. That how you can generate the signal. So this is I already explained you that these are the sources, the potential signal, or like first you need to see the suspicious or case or the cluster. After that, that may be potential signal. You need to redefine that signal. And if it is concluded that yes, it is a signal, then what is your action? And while doing this, you need to consult with the marketing, the patient which is exposed to the whether you have any identify for potential signal, you have any statistical output data or not. You need to consult with the various programmers who are working for these databases. Okay. You need to integrate the information so that you can redefine whether it is a signal or not. Okay. You need to consult with the database. You need to consult with literature whether something is already published or not. What is the background information? If it is something happened with the past as well or not. If we have any background incidences for same or not. If yes, then it may be or may not be a signal. If we have a comparative data, as I told you in the previous slide, that comparator data is very important and it is a kind of source because that comparator, if do not experiencing something and its same is happening with that tester, then it might be possible that that may be a signal. So that is actually manual method, and there is method that is called automated method for signal generation. So same like sources like clinical trial, drug treatment programs, we can signal identifies database with quality and quantitative capabilities. So automated is something which you come to know from any of the clinical trial that this may be a signal. Okay? And what you can do, you can just search based on the query into the databases which are already present because those databases are a repository. Those databases are act as a repository, and you have to just run your specific query to search whether that signal related information is present or not. Okay. Signal verification and information gathering. And after that, you need to assess whether the collaborative decision or veracity of the signal. And this I already told you. Based on that, if you collected that yes, it is a signal, and then what will and you also verified that yes, it is a new signal. Then what is your recommendation for that signal? And not only recommendation or for action, and if you have certain designed for interventions. Like it is a signal, so how can we stop having this signal? And like we need to also communicate that whether the signal is beneficial for us or it is a some adverse effect. If it is an adverse effect, so it taken care as soon as possible. But if it is a beneficial, then in what manner it is beneficial, that need to be again evaluated, and based on that, further action need to be taken. Right. So as I told you that there are some one step like for signal detection or generation automated method hai, and there are individual assetter select those association. So there are certain positive and negative with the association of, okay. So what are the positive criteria? What are the negative criteria? So might be possible if it is unknown area, then it is good. Na? But what if it is known and labeled? Then it is waste of the time. Strong statistical action connections. Okay, it is something which is previously unexpected. Now it is happening. So, but like if we have the weak statistical connection, then no use. Okay, and unspecific tribal events, then also we cannot, it is waste of the time, it is a kind of negative criteria. So these are criteria based on which you can assess your signal as a positive signal. So I will like whether it was previously expected but unlabeled. Now it is unlabeled but it's still happening, then it may be a signal. Okay, some specific characteristics. Okay, specific characteristics are very important. As I told you in the parathnesium example, that nephrotoxicity is only happening in the male patients. So it is specific to the male patient. Okay, and what is the characteristics of those male patient? The age group need to be in between 40 to 60 years. Okay, objective definitive events, typical drug related event or critical terms. So these are something very positive for your signal, and these need to be keep in mind while validating the signals. These are the parameters based on which your signal may be qualified for the positive signal or it is a not a signal actually. Okay. So while detecting a signal or verifying the signal, you need to follow this like low background frequency, serious, high potential relevance. And what are these negative things? Subjective event, common disorder, infectious or endogenous, high background frequency, not serious, low relevance. So these are something very negative criteria for association. Okay. Signal strengthening. So yes, there is a process of signal strengthening. Once we come to know that it is a signal, so it need to be assessed and followed again over the time into the database, like what are the exact number of cases, what are the statistical parameters you have set for searching those reports, what are the exposure of the drug, whether they are just exposed to test drug or not. Okay, the persistence of the characteristics, whether it is persisted for 1 month, 2 month, or 15 days or 10 days. Based on that, we can assess the severity and seriousness of the signal. Consistency of reporting parameters. Okay. For example, if we receive just one email report, then might be possible it is a signal. But if we are receiving multiple reports from different, so it is not a signal. Often, further analysis of the database can be provide the preliminary assessment of the strength of the signal. So yes, it is very important. Important next slides. So the step 3 is like to conduct the follow for that particular signal. If same signal, same database, so you can keep running your query, whatever you have run. Okay. So like to develop over time of the quantitative data of the consistency of the patterns, strength, signal strengthening, the individual case assessment, country comparison, best case, worst case scenario, whether it is a best case, like if you receive multiple reports from your databases. So what is the best case to confirm that it is a signal? And along with that, you need to check which is the worst case. Okay. And how you compare worst case and best case based on the information present in those cases. Okay. Target comparison, nested case control studies, all these I already defined you in the previous slides. The nested case control studies are something that is case control surveillance itself. Signal follow-up in other databases and sources, presence of the similar connections, additional clinical observation, like from if you are looking the same thing in other databases as well, so you need to take some additional information from those databases as well. Okay, because might be possible it is an epidemiological database, so you may get some more information regarding to the epidemiology of the drug. Okay, if that is something ADME drug related database, so you may get distribution resolution histories for that drug. Okay, like what form of drug is causing that? Might be possible tablet is causing that ADR, but the syrup is not causing. So okay. Is that process actually overview of the signal detection process. So this is I already so same sources like these are the sources from where we can get the signal. Environmental scanning, media, medical literature, regulatory agencies, reporting databases, the risk communication. Okay, companies, phase 4 studies, PSUR registries. You need to identify the risk. What is the risk? So signal detection and selection. Once you detect, so what is the risk assessment? Based on that, what is your signal assessment? Once you have assessed the signal, then what is the risk and what is the mitigation strategies for that associated risk? And once you have the mitigation strategy with you, then you need to communicate to those like how you are going to mitigate. You need to change the label. You need to publish the advice, public advisory. You need to publish the newsletters or professional letters, notice to the hospital, press release, media lines, or you need to publish a advertisement or into the various newsletters. I believe that's it. So there are certain challenges in the signal detection. What are those challenges? Just in a two slides. So data quality issues are always there. So like when we are trying to evaluate the signal, so they always have incomplete data. Okay, what happened in the case of pioglitazole and carcinogenic event, and the drug was removed from the market, but because of incomplete data, it was happened, and after some time, that ban was revoked, and the batches of the banned drug were again pumped into the market. Biasness in reporting. So yes, biasness is reporting means if you intentionally want to generate a signal, you might report something which you do not experience, and you can say it is a signal. Okay, so you cannot verify it. So this may be a data quality issue. Regulatory challenges. So global regulations, maybe it is a signal for you, but at global level it is not a signal because they have certain other criteria. Industry standards. So industry standards for signal detections are consistently evolving. Okay, staying compliant with these standards is essential for ensuring thoroughly the safety assessment and the maintaining the public. So yes, industry standards are changing day by day. It are getting evolving. Why it is evolving? Because the global regulations are also getting changes. And why these global regulations are getting changes? Based on their experiences, based on their learning, only they have giving some update in their existing regulations. Okay. So these are also challenges to cope up. Is the challenges. So yes, it is the last one. What is the future trend for signal detection? So like you can see artificial intelligence and machine learning is coming. So as they can have instead of manual judgment or manual verification, AI can do this kind of verification. We have to just give them the electronic query into the format of SQL or something, and you have to feed that query. If something happened, like if something reported in your database related to that, then that can be directly give you a notification that this is an as per your query which you have feed. Okay. Likewise, you may suppose that just let me think. Yeah, that XYZ drug is causing some unknown ADR that might be previously fever was there, but patient getting hypothermic. Fever means increasing the in that body temperature. Now, which was known. Now it is saying hypothermic means the body temperature is getting dropped. So we can make that kind of query that drug causing hypothermia, but it was administered or it is the indication is fever. So if hypothermic as an event will coming, so yes, it may be a signal because we have already assessed it is not published anywhere. So that kind of query you can fit, and if it is happened, so that database itself give you the alert, and it will reduce the manual effort. Big data analytics leverage the vast data sets to be. So yes, like if you have so many databases, so many data sets which need to be evaluated manually, then it is not possible. And same can be done in the single click or two click by these data analytics. Okay. So evaluable regulatory landscapes, so international collaboration. So increasing the international collaboration among the regulatory agencies enables to this sharing. So what I told you that if you cannot collaborate with the other agencies, you cannot come to know what is happening in that country. Okay, and it will not give you like what happened in the COVID. If you were not following the guidelines, if you are not aware about the regulatory guideline of the WHO, that we will not come to know that what happens in the China. Okay, like WHO is the international regulatory authority, and India is also part of those authority, and they are also following the research and the advisory published by the WHO. So yes, they come to know that what happens with the COVID in the China. Based on that advisory, what WHO is publishing on day by day, we have also taken preventive measures for during the COVID. So that is kind of international collaboration. That is the best example of international collaboration. Same way, you can come to know which is unknown in your country, but it is already established in the other countries. Like COVID first come to know first existence. Sorry, guys, I have to share my screen again. So I have shared again. So what I'm saying that international collaboration is very important because what happened, what we have seen during the time of COVID. Okay. And second is the challenges in the policies, changes in the policies. Yes. So whenever there is a change in the policies, we need to modify the regulations based on those policies. And that may we need to ensure that what is now in the policy, like if it was written that previously it was written that only one report can qualifies for the signal, but now if the policy is saying, if the standards is saying that no, we need a minimum of 3 reports to assess a case as a signal case signal, then if you are have the 2 reports, valid reports, still you cannot claim it for signal because now as per new guidelines, you have to minimum of 3 reports. Okay. So I believe that's all from my end for today's session. Yes, guys, thank you very much from my end. If you have anything for any queries, you can ask. Else we are going to close. Any queries from